Yep. So again, welcome everyone to the launch of the Validating Novel Digital Clinical Measures program today. It's great to have you with us here for this event. To begin with, just a little housekeeping. Today's session is being recorded, so slides and recordings will be available after the event. Use the chat box if you've got any questions along the way. And just one other thing, participants are not permitted to transcribe the webinar, so please don't do that. Our agenda for today covers the motivations for our work, the specific challenges that we aim to address, our research and findings to meet those challenges, and most crucially, the resources that DIME has developed for the community to streamline analytical validation and accelerate the adoption of digital health technologies. During this event, you'll hear from myself, some of my colleagues here at DIME, and members of the digital health and statistics communities with whom we collaborated to bring you our new resources. The focus of our work presented here today is analytical validation of novel digital clinical measures. Digital clinical measures are health outcomes or physiological characteristics that are collected by sensor-based digital health technologies, or SDHTs for short. Novel digital clinical measures capture outcomes that have never been previously assessed, either by a digital or a non-digital tool. Analytical validation is a crucial step in evaluating any SDHT as fit for purpose, and it concerns the ability of its algorithms to measure or predict physiological or behavioral metrics. Analytical validation is part of DIME's v3+ framework, which also includes sensor verification, usability validation, and assessing the measure's clinical meaningfulness in its context of use through clinical validation. In the context of specifically analytical validation, a novel digital clinical measure is characterized by the absence of an existing reference measure. At this point, I'll hand over to the CEO of DIME, Jen Goldsack, who will offer some opening remarks and thoughts about what we're launching today. Jen, over to you.
Simon, thank you so much for welcoming me to the event today, and I really am excited to be here. This is such important work, and congratulations to everyone who participated in this effort for putting out these resources today. You know, when I think about the work that we've done here at DIME as it relates to digital measurement, and as it relates in particular to the use of digital clinical measures as endpoints in clinical trials, we've really proven out, for example, with our partners at Tufts, the value of these measures. And if we really want to take value in its most sort of basic form, we know that there is up to a 600% multiplier on the investment in the use of a digital endpoint when it comes to calculations of estimated net present value. Really, that translates into hundreds of millions of dollars of value being conferred upon a clinical trial, reductions in trial timelines, enormous benefits by being able to measure some of the most valuable facets of disease, yet we haven't embraced that sufficiently because those most valuable measures, those measures that we've struggled to capture previously, those measures that when we talk to patients and their care partners, they tell us are the most important symptoms or characteristics of their disease. When we talk to scientific decision makers, when we talk to regulatory decision makers, payer decision makers, clinical decision makers, we learn about these characteristics of disease that are critically important, but that we've struggled to measure previously. That's the power of these novel digital clinical measures. And yet we have struggled as an industry to really bring them to scale in medical product development. And it's a very sort of methodological issue. And that's the issue that we've addressed during the course of this project. We've been able to think about, well, if the value of the measure is that it's a facet of disease that we haven't been able to capture before and yet is inherently meaningful, how do we actually know that we're measuring what we're claiming to be measuring? How do we select an appropriate reference measure or basket of reference measures, knowing that it's not quite right, because the concept we are probing by definition is truly novel? And then once we've decided about what that reference measure is, or that basket of reference measures, how do we think about the appropriate statistical methodologies to make the crosswalk between what we're measuring de novo for the first time using sensor-generated data and that facet of disease that patients, their families, clinical scientists, clinicians, regulators, payers have told us is most important. That's the work that we've done throughout the course of this project. And it's so important because it really is one of the last scientific bottlenecks in fully capturing the value of digital clinical measures as digital endpoints. And this has been a bit of a different model for us at Dime. We recognize that if we want to do some of this more methodological work, we have to get creative in how we make it happen. So with that in mind, I want to thank not only Simon, you and the project team, you've done such a tremendous job, but I also want to thank our funders at Arnold Ventures for recognizing that if we could unlock this methodological bottleneck, we could capture so much value for industry, for the process of drug development, and most importantly, for the patients that we're here to serve. I also want to thank everyone who served on our statistical advisory committee, many of whom are experts on the line today who you're going to be hearing from, and in particular, our colleagues at FDA, and Andrew Potter in particular. You've been a brilliant partner in this, our official liaison, and really grateful for your input and insights onto this. So with that in mind, I'm excited to share the best practices that we are recommending in terms of how we as an industry can capture the full value of truly novel digital clinical measures. And with that, Simon, I'm going to hand it back to you.
Thanks, Jen. Let me just get the slides back. Yeah, thank you. That's for those really poignant and insightful comments about why what we're doing here today matters. And yeah, a little bit about the setup. So the resources presented here today are the culmination of 15 months of collaborative work between Dime and the digital health community. And for this effort in particular, as Jen mentioned, we convened a statistical advisory committee, which is a curated group of statistical and digital health experts with individuals representing the diversity of our field, including representatives from the FDA. And the committee supported the development of the resources, and you'll hear from people in the committee today later in the event. And as Jen mentioned, Arnold Ventures were the funders and was super grateful for those, for supporting our work. So the resources you'll see today will support digital health community and their analytical validation needs in several ways. The framework for validating novel digital clinical measures provides a systematic and standardized approach to conducting a rigorous analytical validation study. The framework covers 4 key aspects of study design, which you can see on the screen there, and it's nice and clear and brief. The framework itself is implemented in 2 different resources, which allows digital health technology developers and biostatisticians to choose the most appropriate way to gain value from the framework and its principles. First, the interactive guide to validating novel digital clinical measures provides a handy and dynamic quick start guide to the framework and its principles, walking DHT developers and biostatisticians through the points on the screen there. Secondly, Build Your Study, Novel Digital Clinical Measure Analytical Validation Planning, or the Study Builder for short, combines the framework's principles with other key Dime resources to provide DHT developers with a practical step-by-step guide to designing a rigorous study for their measure of interest. Finally, the Simulation Toolkit for Digital Clinical Measure Validation contains a selection of materials that complement and support the other resources. And amongst the things it does, allows a researcher to explore a method to assess relationships between measures with non-comparable units using a simulated analytical validation study, and then tailor an experiment with model factors to simulate scenarios that are tailored to their particular scenario. With that brief introduction to the resources out of the way, I'd like to move on to our first panel of the day, a discussion on the current state and future of novel digital clinical measures, the challenges faced by the community with regards to their validation, and why our efforts here today are exciting in how they address those challenges. Our panelists, all part of the Statistical Advisory Committee, are Bogdana Ratic, Science Fellow at Bayer, Eric J. Daza, founder and chief editor of Stats@One, Joss Langford, CTO and founder of Active Insights, and Lisa Martis, Director of R&D at Cove Neurosciences. Eric, over to you to moderate the discussion.
Great. Thanks for the opportunity to moderate and be involved, to you, Simon, and to Dime. So I've worked as a biostatistician and data scientist for over 21 years in industry and academia across various biomedical, clinical, and health areas. I also created Stats of One, a newsletter and podcast on digital health N of 1 statistics frequently found in N of 1 trials. So I was excited to join this Dime project because the standardized approach that we're creating to validate DHT-derived measures really provides this strong foundation to build digital endpoints. So let's first revisit what Simon told us about novel digital clinical measures. They capture health outcomes or characteristics of an individual's condition that, one, haven't previously been assessed, two, are collected by sensor-based digital health technologies, and three, lack an existing high-quality reference measure. Let's go to Leif first. Leif, you've been working in some pretty exciting areas of novel digital clinical measures. Please introduce yourself and talk a little bit about the sorts of novel measures that you've been working on, and how do they do things that just really wouldn't be possible without working in a digital space?
Yeah, thanks so much, Eric. So I'm Leif. I'm a researcher at University of Toronto and also director of R&D at Cove Neurosciences. So I have a background in, implementing, developing novel digital measures for all kinds of, neurological and neurodegenerative disease indications. From my perspective, there's been this really massive push quite recently to make use of meaningful digital measures. And one of the main stumbling blocks that we've kind of come across is being standardization and developing meaningful frameworks for practitioners to operate within. Dime has been doing great work in this area. And when I learned that there was more effort being taken to extend and build upon their work, I was really excited to get involved, especially given how impactful their work has been for my research and my industry work so far as well. For example, my academic speech research, it was very important to be working in a digital space, especially in diseases like ALS, so that we can enable scalability and access both for researchers to a diverse group of patients and also for patients to access care. And digital technologies really enable this enhanced combination of scalability and novelty in the measures we can extract that otherwise wouldn't be possible without digital technologies.
Yeah, thanks, Leif. That really shows what the power is, right, of novel digital measures to do something that's incredibly important, like advancing our understanding of ALS and other diseases. And these things just wouldn't be possible outside of the digital health space. I particularly love your summary of their value as objective measurements, right, of novel aspects of function that are done efficiently at scale. Very cool. So turning to you, Joss, you spent many, many years working hard to develop digital health technologies and pave the way for breakthrough advancements in clinical care. What are some of the values that digital measures can bring to a clinical setting? And what are some of the main challenges involved in validating their algorithms to actually get them there?
Thank you, Eric, and thank you to the team at Dime for the opportunity to be involved in this work. I specified my first accelerometer back in 2002, so I've been in the space for a while. In 2009, we founded Active Insights with the original raw data wrist-worn accelerometer, and much of that early work has been in public health. And in 2021, I had the opportunity to be involved in the Dime Digital Measures Playbook, which really brought these technologies firmly into clinical setting. That's sort of a bit of the history there. Adding to Lee's point about objectivity, this is critical as it helps us to understand and control some of the biases that are associated with more subjective measures. I think one of the benefits these bring. Digital measures also provide a basis for a more patient-centric perspective, continuous monitoring within the patient's everyday lived environment, and often with lower cognitive burden. And from a measurement point of view, they hold out the promise of much greater resolution and sensitivity than traditional measures. And this, as I think Jem set out at the beginning, is the basis for helping us reduce costs and timelines. Some of our challenges around validation include ensuring there's a clear conceptual distinction between the sensor technology capturing the data, the physical product that embodies that sensor, and then the algorithm that converts that data into meaningful measure, and standardization is important there. For us, selecting an appropriate reference measure is as much of a challenge as methodology selection. And we're always working with our clients to understand the context around a measure. What may be well understood in one area is often novel in a new area.
Yeah, thanks for that, Joss. I really like this distinction that you make between the sensor-based digital health technology as a whole and the product that it sits in, and also how the algorithm specifically converts the sensor data into a meaningful measure. Analytical validation focuses on the latter, which is what we've been working on in this project. So it's clear from what you say that the huge potential of novel digital measures is blocked a little bit by lack of standardization around validation. But Donna, this is a great place to bring you in. First, please introduce yourself and then tell us about the importance of standardizing approaches for validating novel digital measures. And why are you excited about the work being presented today?
Hello, and thank you very much for the opportunity to be part of this event. I'm an expert statistician at Bayer. And in the past few years, one of my focus areas has been on the use of DHTs in clinical research, and most of that work was on statistical methods for clinical validation. Some of it was in collaboration with DIME, and so now I'm very happy to see that DIME again is leading the work on the framework and statistical methods for another validation stage, which is analytical validation, another foundation on which the clinical validation can build afterwards. So from my perspective, this type of work is important for at least two reasons. On the one hand, there are already a lot of validation methodologies and tools that exist in other areas, such as validation of wet biomarkers and payors. And so they should be brought into the DHT community so that we don't reinvent the wheel. But on the other hand, validation of DHTs has some unique challenges. So one needs to adapt the existing methods to this context, and to do that with a lot of care. And as others already mentioned, some of these unique challenges stem from the fact that we have a potential of measuring with DHTs very novel outcomes and in very novel measurement conditions. And so this project is addressing some of the key challenges, and namely situations where there is no obvious and directly comparable reference measurement method. And it's really nice that the framework that is released today defines this spectrum of scenarios with different types of reference measures that one can conceive of and really utilize in various ways. The study builder and the simulation toolkit is also a great resource, and including for statisticians for planning an analytical validation study. It will allow analysts to assess the potential impact of measurement errors and to some extent patterns of data missingness. These are very important aspects to take into account, especially as DHTs are intended to be used in 3 living conditions. So these factors can vary a lot and need to be accounted for in the plan. So it's great to see that this team raises awareness of these issues and also provides some tools to work on them. Thank you.
Great, thanks, Vadana. I really like how you situated this work on this project, right, within the wider world of validation methodologies applied to lots of different areas. But you also highlighted what's unique about DHT analytical validation. So there's a clear advantage to standardizing validation methodology, and it's great to hear that what DIME is doing here will benefit the community. Okay, so in summary, novel digital measures hold huge promise to capture aspects of health that were previously impossible to identify and measure, but challenges around their analytical validation exist. And having a standardized approach to validation, like the one DIME is presenting here today, holds immense value for the digital health community. I want to thank all the panelists for that very stimulating discussion on the state of novel digital measure development. I'll hand back to Simon now to talk about the work DIME and the advisory committee have undertaken over the last 15 months.
And thanks to the whole panel for that discussion. That's really interesting stuff to hear there. I think the key takeaways, as Eric said, is there's great opportunity with novel digital clinical measures, but there are challenges around particularly reference measure selection and also appropriate statistical methodology to use in analytical validation before we can get the most out of the potential they offer. So this is really what we and DIME and the Statistical Advisory Committee set out to do over the last 15 months. And in particular, the two things that we set out to address are systematic criteria for selecting reference measures when undertaking analytical validation of digital clinical measures, and secondly, how to assess relationships between novel digital clinical measures and reference measures when the units are not directly comparable. And we'll come back to that a bit later. So with that defined, let's now talk a little bit about the approach we took to address the first of these challenges, starting with that first one, systematic criteria for reference measures. At this point, I'm going to bring in a couple of my colleagues, Andrew Potter, Senior Mathematical Statistician from the Office of Biostatistics in FDA's Centre for Drug Evaluation and Research, and Ben van den Driesche, Chief Delivery Officer here at DIME. Ben, if I can turn to you first.
Yeah, absolutely.
Yeah,
absolutely.
Just tell us quickly who you are and your role in this work on reference measure research.
Will do. And just want to call out that I'm really happy to be here for this launch event. By ways of brief introduction relevant to this work, I did a lot of work on analytical validation before it was called that, and ran into many of the bottlenecks and challenges that we've tried to address with this work. So it's really, it's been really fun to apply that past experience to what we're launching here today.
Yeah, right. Thanks for that. Briefly, if you can, could you take us through the key findings that DIME's made here that's going to help the community with their reference measure selection?
Yeah. So like was clear from the previous panel in the introductions, there's definitely many situations when you need to conduct analytical validation for an algorithm that may be relatively straightforward, right? Because there is already a well-established field. There are reference measures available, a lot of prior work out there that, puts that in perspective. But then when you talk about these novel measures, that is definitely, that work is simply not there, right? So you don't have that base to fall back onto. And as already pointed out previously, these novel measures really have the potential to have an outsized impact on scientific and clinical decision-making, find cures for more complex conditions that we're all collectively working on. So to accelerate their adoption, we need a way to make that analytical validation process more streamlined for everybody involved, from developers to trial sponsors to regulators. And to deal with that, we developed a reference measure framework, which specifically encodes that not all reference measures are created equal, right? Depending on your context of use, the rigor of your measure, some choices may be appropriate and some not. And having that conversation, going through that thought process is not necessarily easy. And the framework helps to streamline that decision-making process and give us all something to hook into and have and have that conversation and do that planning. In the work, and it's going to be linked in the chat here, we identified two categories of reference measures. There's objective reference measures, which, as the word implies, they measure through objective means, meaning they do not rely on human observation. And then you have observed measures, which do rely on human observation. And then we have additional subcategories that become more granular in those. And as we talk about hierarchy of such a framework, the objective reference measures, or if available, are always preferred because of their increased rigor over an observed measure. But depending on your context of use and other considerations, such as patient safety, for instance, they may be appropriate, it may not be. So really, the framework tries to capture all of these different nuances and how to approach them when you think about identifying a reference measure.
Yeah, great. Thank you for that summary, Ben. That's really, really clear on the general way the framework works. But to dig in a little deeper, it's clear that there's a clear hierarchy here for reference measures and selecting them based on the rigor of the reference measure, and that's going to really help an investigator make an informed choice. But in the novel digital measure scenario, particularly, reference measures may not exist at all, or they may not be very strong at all. So specifically, what's the framework offering in the novel scenario, in addition to existing reference measures?
Yeah, so what I was describing just before is really the scenarios where you're trying to identify reference measures that already exist, right? And to your point, if you're dealing with something truly novel, they may simply not exist. So also here, the framework also captures that in that same hierarchical approach, in the sense that we defined two types of what we're calling novel comparators that can be developed specifically for an analytical validation study, right? And a novel comparators is exactly that. It's a reference measure developed from scratch because an existing reference measure is simply not available. And they are things that can be observed and counted by medical professionals or self-reported by a patient, right? They're not going to be objective because we are lacking another piece of technology that can measure that thing and that has been properly evaluated previously. So that's one category, but then that in itself may also not always be possible, right? There are cases where you may be prevented from doing that. There's simply, it's so novel, it's such a specific context of use that even that doesn't work out. And then there's a final step in the framework that relies on anchor measures. So an anchor measure is a measure of physiologic process or behavioral construct, just like any reference measure, but in units that are not directly comparable to your measure of interest. So because of that, from a statistical point of view, you're limited to examining associations. There's no way to look for direct correlation, right? So this is really a last resort because technically speaking, this is not true analytical validation anymore, right? Because you don't have that ability to look for direct correlation. And because of that, a single anchor measure is probably not going to work. You're probably going to need more than one and really look for association between a number to try and come up with some data that your algorithm is in fact doing what you claim it's doing.
Yeah, no, exactly. Yeah, that's really the key thing when you get to that situation that yeah, picking multiple anchors is really going to help validate when you really run out of those options. But yeah, it's clear there's, you know, through the framework, you know, it's obviously broadly applicable to analytical validation at large as well. But it really does obviously support the novel situation with those comparators and measures options as well, anchor measures options. So quickly, you know, the novel digital measure world is obviously, you know, the options and possibilities are endless. You know, how does the framework allow for an investigator or a user to kind of leverage their specific subject matter knowledge in that novel world and kind of use their knowledge?
So I think, I mean, it's really kind of like from a more of a futures perspective point of view, right? Like, let's say that you've got a measure of interest and the only thing you've got available is a self-reported reference measure. You will need to do quite a bit of work to justify that reference measure is appropriate for your context of use and that the framework will help you come to that conclusion, hopefully. By doing that work exactly, you're actually establishing another piece of evidence, right, about that measure of interest. And maybe a future study can expand on that and look for a different intended use environment or patient group or whatever it is that you're trying to accomplish. And ultimately, what you're doing is you're upgrading your reference measure, right? As you look at the framework and the hierarchy in it, you're literally like moving up in the steps and establishing that reference measure as something that has more rigor. And collectively, as a field, as this information becomes available, that is really the driver of this adoption, right? Because collectively, we raise the bar about where the level of evidence is and what type of claims can be made about a specific measure of interest because we have more robust reference measures. So a lot of the work that DIME does, as many people here are familiar with, is focused on generating these shared resources and frameworks to make sure we all speak that same language and we can build on the work that was previously done instead of collectively feeling like we need to reinvent the wheel over and over again, right?
Exactly right. Yeah, building shared resources and that shared language is super important and a super important part of this framework as well. Ben, thanks for that and taking us through the reference measure framework. Andrew, if I can bring you in here at this point. So first of all, like just before we discuss the reference measures, could you just tell us a little bit about your role in the advisory committee and specifically as a PPP liaison from CEDA?
Yeah, thanks, Simon, and thanks, everyone. This is a great event so far, and I've been very happy to be involved in this work. So I'm Andrew Potter. I'm a statistician at FDA's Center for Drug Evaluation in the Office of Biostatistics. I've been doing a lot of work with, digital health as well as neuroscience applications, and there's a lot of, so hearing all this stuff about objective, subjective, from neuroscience, psychiatry, I go, wow, this is, exactly what we've been trying to work on in endpoints there as well. So, but public-private partnership or PPP liaison is, so FDA can join, with outside groups such as DIME or other groups, a public-private partnership, and then have official liaisons for work that's important to FDA's mission, and allows for FDA to, so we can actually participate in this work, that we know kind of what's going on, and we have an official sort of format for participating where we don't, where we're getting something we want out of it, and that it's available to the, to the entire community, and there's kind of formal ways to make sure that we do this so it comes out to everybody and can be used by everybody, and allows me to give feedback, and it allows sort of as the agency to give feedback. So hopefully that helps a little bit.
Really does, and it's been great having you involved formally with the project, and thanks to you and to your department as well. So briefly, the reference measure framework. How do you feel that reference measure selection, having a framework for that, is going to be valuable to you and your colleagues?
It's going to be very valuable, because one of the hardest, anytime we're starting to look at, a DHT, some sensor-based DHT measurement, even if it's a measure, even if it's not super novel, there's always this question of, so how do we have evidence that's measuring what we want? And we know that there's going to be, for example, with steps, we can go put someone in a lab, have them count steps, but really trying to say, for that, how good of a measure is our existing tool, so we can say, okay, yeah, we've got a really high quality measurement conducted in a clinic, maybe, that is, that we can look at some of those methods that say, hey, do these two pieces of, do these two ways of measuring steps correspond? Or, maybe as we get a little further away from steps and it's looking at, daily physical activity at home with subjects, that's going to be a lot harder. Maybe we can have some good self-report. Maybe there's some good clinical questions, I'm going to say clinical anchors that can be used. But we have now a structured way of thinking of that. We can start to think about it of, how do we think of these reference measures? We can link it to some of our patient-focused drug development guidance, which also talks about how do we validate measurement, how do we build evidence for the use of, primarily COAs, but sensor measurements can overlap with both COAs, biomarkers, something completely new, and it gives us a way to really start to think through, hey, we've got this existing guidance, we've got all of this existing stuff, and now we have a way of saying, hey, here's some ways we can start to organize our thoughts on reference measures. And then importantly, when it comes into the agency, it's more organized, it's easier for us to actually find the information about reference measures, and we can say, hey, it's here, great, we can review it, or it's not here, what about this? We can directly ask for it, and hopefully there's a nice shared language that we can all use, so we can clearly ask about it and clearly know, is it coming in, and know when we actually have the information we need.
That's really great to hear, and that shared language is what we've tried to get, and be quite rigorous and define things through this framework. So thanks for that, Andrew, and thanks also again to Ben for that that stuff there. So that covers the first of the two questions that we look to answer with our work, and the second of those two questions is how to assess relationships between measures when units are not directly comparable. So briefly, units are not directly comparable in our definition if they are directly comparable if they are identical or can be translated for the purposes of comparison, and they're not directly comparable if that isn't the case. So, this is a challenge that's not just about units. This is about statistical methodologies, and really just, lots of techniques are employed in validation studies, but not necessarily appropriately. And where you don't have good reference measure choices, techniques are not great either. It's hard to be confident when you validate. So let's quickly jump over to our next panel, which is to talk about our research approach here. We've got Birand, we've got Chen, and we've got Colleen, who are going to be talking through our work and our approach here. So Colleen, if I can jump to you first, can you just briefly tell us why you're excited to join the effort first?
Sure. Thank you, Simon. I really appreciate being involved in this committee. I've been working in the drug development and clinical trial space for some 30 years, but about 12 years ago, I had my first clinical trial that had a sensor technology in it. And I remember distinctly thinking, hey, this is the future. And so I was really excited to join the advisory committee and to work with you and the team. Because, I recognized that there was, a need for more guidance for this very important step of the process, the analytical validation step, and just the need to address the science and the statistical methodology and, define really more closely, what does this evidence really actually look like? And what would be some useful tools to answer that question?
It's been great to work with you on this effort. So, very briefly, can you tell us why using appropriate techniques in validation is important?
Well, I think manufacturers of the tools and researchers themselves for the novel biomarkers work really hard to establish the credibility of the tools and the endpoints, and really to build confidence for the users, and also, to just promote wider use, of course. But I've found that sometimes the appropriate methodology is not always applied, and there's a tendency to not consider all the factors that go into how to define a trial for analytical validation. And the example of what you just cited earlier about the use of the units being on different scales is a completely different question and a way of framing your study, as opposed to a situation where you would have two things that you're comparing that are on the same scale. And so not only the question changes, but the statistical methodology changes as well.
Exactly, really personal observations there, and I'm going to have to move on because of time at this point. But
Sure.
yeah, you make a really good point that analytical validation with novel digital measures, and selecting reference measures with our framework that we've just shown, a researcher is going to often find themselves, in that situation where units are not necessarily comparable. But they might be using reported measures such as patient-reported outcomes or clinician-reported outcomes. And those units won't necessarily match.
Mm-hmm.
And ways of addressing assessing relationships are pretty limited in those situations usually. But what we've taken as an approach, and Birand's going to come in and talk about to set the scene here, is a new approach potentially to assessing relationships that's motivated from those patient-reported outcome fields, which is confirmatory factor analysis. Birand, if I can quickly just ask you about factor analysis. This is something that comes from psychometrics, the field of measurement in psychology, right?
Yes, that's right. Traditionally, exploratory factor analysis was used to understand the variability among a set of correlated variables, such as the items of a questionnaire, as the expression of a smaller number of underlying factors. And in confirmatory factor analysis, we already have an idea of the underlying factor or factors, and the analysis focuses on the relationship between the factor or the latent construct and their indicators or items.
Yeah. Thank you for that context, and that latent construct point is really key. Maybe with digital measures in a validation scenario, you don't have comparable units, but maybe they're both capturing part of the same latent construct. Can you briefly give a quick example about latent constructs?
Yes. For example, think about depression. We cannot directly observe or measure depression, and therefore we say it's a latent construct. Yet we know it exists because we can infer depression from observable indicators such as a low mood, fatigue, loss of joy, poor sleep, etc. So a depression questionnaire consists of a number of items asking respondents for those symptoms that we assume are associated with depression.
Yeah, thank you for that context. And I'm going to have to move on here, but that context about latent and observable indicators is really key, because that motivates the idea that if two measures are capturing the same latent construct, say depression, then even if they don't have comparable units, then we can actually use CFA to assess correlations between the measures using that idea. Thanks, Biren, for that. Chen, if I can briefly come to you. So we worked really closely on our simulation study work. And yeah, so for context around that, DIME investigated this idea for CFA and its appropriateness in a validation study by using a simulation study to characterize its performance and behavior. And then we validated that approach by applying it to a number of real-world digital health data sets. So Chen, yeah, thank you for joining us here today, and also thank you for coming to say some words about the simulation. If I can skip quickly to the discussion of the simulation, we worked really closely on what was going there on the work there earlier in the year. Could you quickly just kind of really briefly just kind of summarize the results of our simulation study and just let us know a little bit more about that?
Yeah, sure. Thank you, Simon. For the simulation study, we, as you mentioned, we introduced CFA and together with another regression method, and we compare that with Pearson correlation analysis, which is the traditionally used statistical method to assess association. And the first thing we assessed was feasibility, whether our CFA models in the scenarios converged. And what we discovered was in majority of the scenarios, it did converge, and it has acceptable fit statistics. And the second part we assessed was the statistical properties such as bias and variations. And what we discovered was that the CFA methods is in general less biased compared to Pearson correlation as far as recovering the true relationship is considered. And this, however, it's worth it to call out that CFA method is less stable as compared to Pearson correlation. The other caveats that we need to pay attention to is that the CFA method typically requires a well-sized study. However, this, as we discover in the real-world data, is that this can be compensated by adding repeated measure in our study design.
Thank you, Chen, for that summary. And sorry, we don't have more time, but yeah, I think that's really good to hear that CFA is potentially a new tool that researchers can use. And it's feasible in validation studies. There are some challenges, but that's the case with any method. And with good study design, you could potentially mitigate those and really use it as an extra tool in your validation arsenal. Thanks everyone on the panel for chipping in on this simulation study and statistical methodology discussion. At this point, I'm going to switch back and talk about actually how our research findings came together and talk a little bit more about the resources involved. So as mentioned earlier, the framework for validating novel digital clinical measures is a systematic and standardized approach for conducting a rigorous study of a novel digital measure. And it takes a DHT developer or biostatistician through its four key aspects. There are two different resources implementing that framework, and we're going to start by taking a quick look at the first of those, which is the interactive guide for validating novel digital measures. So this is it's a high level, this framework, this interactive guide presents a high level decision process for designing an analytical validation study in an interactive and dynamic format. It introduces the structured framework that has just been mentioned just before this, with selecting fit for purpose reference measures and designing a rigorous study, compiling evidence to justify claims, and also the important first step of defining your objectives correctly. You're able to click on various different icons in the framework, and also the rest of the guide, in order to get further explanation about things, such as here, where you're invited to seek further regulatory interaction. It's really easy to navigate through the guide, straight to a section of interest, or just to work through the guide page by page and find all the information in a really easy to understand format. There are links to various different other resources that are helpful at various different stages, such as the DINE's V3 framework, and also the other resources from the project, which are the study builder and the simulation toolkit. That's a really quick look in at the interactive guide. And now let's have a quick look in at the other framework implementation, which is the study planner. So this is much more practical of a tool, and it brings together not just the reference measure framework and the recommendations from our simulation study, but also existing DINE resources, and it combines it into a step-by-step guide for planning analytical validation of your digital measure. The study builder is split into 6 different sections, and users can work their way through the different questions. They can get plenty of different information in terms of links out to other DIME resources, clarifying information, or other things such as extra information to give users complete confidence to answer their questions. Users can enter their answers in the boxes shown here, and download a partially completed or fully completed document to continue working on or distribute with colleagues. At different stages, people are encouraged to either check and justify what they're doing to make sure that they don't miss anything, or to have a hard stop and not proceed any further if there are, based on particular conditions. And the end of the guide is a handy checklist, which means that people, when they're compiling their dossier of evidence to support analytical validation, they really don't miss a step. And this study builder will help you standardize and streamline the process for your analytical validation study and take the uncertainty out of your regulatory interactions. So that was a very brief guide to the 2 resources that implement the framework. If I can share my screen, then we're going to have a very, very brief discussion about the resources that we just saw. And that's going to bring back in Andrew Potter from earlier, and also Joss. So, Andrew, if I can quickly go to you, can I just get you briefly to say, what do you think about these resources? How do you think they'll support the community and and yourself in regulatory situations?
Thanks, Simon. Yeah, these are great resources, and it gives us a place where we've got a common language, a common kind of set of steps, so we all know, hey, here's where we want to start, so we can get something on analytic validation, start building evidence, and see where it goes, and give us a way where we can also come back, revisit how all of these tools, and make sure, hey, they are working, they're doing what we want, and we can do that in a structured way, and in hopefully a standardized way, so we're not having 40 different developers and pharmaceutical companies doing it 50 different ways, which unfortunately we see. So it helps both on the community end, and then on our end, kind of we all know where to look for the same things.
Awesome. Thanks, Andrew, for that perspective. And it's really good to hear from the regulatory side that what we've built here potentially has value in standardizing submissions and making your life easier. Joss, if I can bring you in now as someone on the other side of the coin, how valuable do you think the interactive guide and the study builder are for people in your situation?
Yeah, thank you, Simon. So looking from a perspective of a developer of DHTs, the interactive guide it brings together many of the resources already developed by DIME, and you've seen these being posted in the chat, and I do encourage you to go and look at those. It puts them in context of a directed plan and the jobs to be done in sequence. And I think that's an invaluable project planning tool. It'll help to identify risks, uncertainties, and tasks to complete. For me, the study builder then really is a framework for collecting the evidence, not only so that you do that in a consistent way, but also giving yourself confidence that you've done it comprehensively, because the risk is always you don't know what you don't know and what you've missed. And I think this framework will really help with that. So overall, whatever your view on standardizations of the digital measures themselves, standardizing how we describe them has to be the right thing to do. As a community, working with the regulator, having an agreed language, nomenclature, and structure and for our work has got to make things easier. And novel measures are difficult enough without having to deal with novel descriptions and processes every time.
Yeah, exactly, indeed. Thank you, Josh, for that, and thank you, Andrew, for your contributions there. So that's the two resources that implement the framework. And I'm going to briefly talk about the third complementary resource now, which is the simulation toolkit for digital clinical measure validation. So yeah, let me just take a quick look at that. So that toolkit can be found on the project webpage or through any of the other resources. And it includes in-depth resources to evaluate new approaches for comparative analysis when the units of your digital measure and your reference measure are not directly comparable. The primary component of the toolkit is a code repository hosted on GitHub that you can see here. And it contains everything a biostatistician is going to need to investigate the performance of confirmatory factor analysis and other common methods when assessing relationships in novel digital measure scenarios is what's on the table. You could start by replicating our original simulation study and just running it on your device as is. And you can also, access key visualizations to understand and explore the characteristics of the methods. The code is fully documented and commented, so you can get straight in and dive in straight, quickly and in with confidence. The documentation also tells you how to experiment with the factors of the simulation, so that you can really tailor it to your scenario as well. And that's a really nice feature. You can understand, in your digital clinical measure scenario, how the different methods might perform and how CFA might help you out there. In addition, we've got a detailed statistical note about methodology that's relevant for not just novel scenarios where you don't have comparable units, but also for a broader range of scenarios as well. And you can also access the manuscript underpinning a lot of this information and get access to the full simulation results and the full theory and content there. So that's a very brief look at the simulation toolkit. I'm really excited for this because it's the first time that DIME has offered a code repository to the community. And just to briefly talk about the value of this, I've got, again, a couple of quick comments, again, from Andrew, and I'm going to bring in Libby Floydeen for the first time, who's head of endpoints and a biostatistician at Evernova. Andrew, again, if I can just really quickly turn to you with some really pointed comments. How important is a code repository for the community to have here and to work with and build upon as a stepping stone into understanding new methodologies here?
I think it's hugely important, because now, instead of having to recreate the code, ask, have we validated our software, we've got one place that we can pull up the software, say, hey, here's the work that's been done to validate it, here's how it works, and we can jump into it and start applying it to our situation. It also makes it easier on the regulator end, because I can go grab the code myself, start experimenting with it a little bit, get some familiarity with how these methods work. So as they come in, I'm not starting from ground zero. So I think, very, very important to have.
Yeah, thank you, Andrew. And thanks for your comments all the way through this session. Libby, if I can turn to you now, just briefly, just tell us a little bit about why you're excited to join this event before we talk about the toolkit.
Yeah, thank you, Simon. I lead the endpoints team at Evernova, and we're a health tech company that's part of the AstraZeneca group, and I'm a biostatistician also by training. And I was thrilled to be a part of this advisory committee and contribute to this work alongside so many talented individuals. But most importantly, to be able to put forth an open source tool to facilitate digital measure acceptance, essentially.
Thank you, and it's been really great to work with you throughout this whole 15 months. So yeah, briefly from your perspective as a biostatistician, how do you feel this toolkit can add value to the community? Maybe how do you see using it yourself?
Yeah, well, in general, I find data simulation an incredibly helpful tool, and both in understanding the boundaries or the limits to a statistical method in a particular situation or set of situations, but also for study planning. And so personally, I'm excited to use this more so for the study planning purposes, right? So when thinking about a validation study, it is so helpful to be able to understand how the statistical parameters of interest are affected by the digital measure and the different characteristics of your reference measure. So I expect that this will take some of the guessing and, and the finger crossing out of this process. One aspect that is particularly helpful is seeing when a confirmatory factor analysis can be leveraged successfully for this purpose. This methodology has been challenging for me in the past. Chen briefly talked about issues of convergence, for example. So by using this toolkit, I'm able to recreate similar scenarios using characteristics from real study data and tweak it such that I'm able to understand what is going to be needed for success.
Awesome. That's really great to hear. And it's exciting to hear that there's value here for biostatisticians such as yourself, and that you're potentially getting into it after this session. So thanks for that. With that, that's the end of that panel. Thank you for everyone contributing there. And really, that leads me to wrap up the entire session and to say thanks to everybody for joining. It's been really great to share the resources with you today, and to share the interactive guide, particularly with giving you a clear decision process for your study, taking the uncertainty out of interactions there, and the study builder as well, offering a really step-by-step guide through that process for building a study for your analytical validation needs. That's been really exciting to share, and I really encourage everyone to take a look at the resources in more detail after this session. So as we draw to a close, thank you to all the speakers here and moderators today, and we look forward to collaborating with you again very soon. Just a couple of quick announcements before we finish up. We have an event on December the 12th later this week that you can see more details about how to register for in the chat. We also have an event on pediatric rare disease that you can also join us for later on. You can use the QR code there to register and to join us. And we also have an announcement about diabetes management and driving market access for that. And again, you can join us for that session, and you can use the QR code shown on the screen to get more information about that. So with that said, thanks to everybody for attending this event today. I hope that taking you through those resources has been an illuminating process, and that you can take those, explore those further, and take them into your work to accelerate the adoption of novel digital clinical measures in clinical research, and really take them forward and do some great stuff with them. We look forward to seeing you again very soon. I hope you've enjoyed this session. So with that, thanks again, and goodbye.